Hi everyone, this video is about homologous genes and chromosomes. Okay, so we've been kind of saying some confusing stuff, and the point of this video is to really try to sort this out. So we've been saying things like the same chromosome pairs will pair during meiosis, the pair of the same chromosomes, right? We've talked about same and different chromosomes. But then we say, well, the chromosomes aren't the same. In fact, we're interested in their differences which are there due to mutations. So then, in what sense are they the same? And the other side of that coin is, in what sense are chromosomes that we do not say are the same, in what sense are those chromosome pairs not the same? So this is pretty confusing, and the point of this video is to go through a toy example to try to clear this up. Okay, so let's imagine a new island or a new planet or a new place that is colonized by one single haploid cell. And we're gonna draw this cell a little bit differently because the screen's gonna start getting filled up with them, so we're gonna draw it like this instead. So here we have two chromosomes, and let's point out that this would mean these are different chromosomes, these are different collections of genes. Like maybe the pigmentation gene in the species is up here, maybe lactase is down here, and maybe the gene for NOVA is here, and a ribosomal protein coding gene is here, right? This black chromosome has a set of genes that are of totally different sequences and functions to the set of chromosomes that are linked on this the set of genes that are linked on this blue chromosome. Okay, so we start out with these two very different chromosomes, and we start with our new population, our new island, with a single haploid cell. So over time, that haploid cell is going to divide by mitosis to give rise to identical cells. And so what we're doing here, notice, is that we're gonna say that the past is back here. So this is the beginning of the population, and that time is going forward as we go to the bottom. So this was the first generation. Now this is the second generation after this one is divided. So that means that these two cells live at the same time this cell lived a little before. And then these two cells are going to divide, and now we have, in this population, at this time we have 4 cells. And then we have 8 cells. Okay, so during these replications, very occasionally a mutation is going to occur. So here where I'll use this little notch to indicate a mutation. And so let's have a look at that mutation. So mutation seems sort of mystical. It's worth pointing out that a mutation is just a change in the double-stranded DNA. So if we just blow up this tiny little bit of chromosome and look at about 100 nucleotides, we have some sequence. And then when we say that there's this mutant copy, all we mean is that there's been a small change. And in this case, it's just a single base pair. See, right here, we used to have an AT, and now we have a GC base pair. So one important thing to point out is that a mutation is not weird. It's not identifiable. You can't look at the chromosome and say, oh, there's a mutation. It's only by comparing between different individuals in the population or between species that you can say that a mutation has occurred because this is different than this. That's the definition of a mutation, a stable change in DNA. But you can't even really tell which one is the mutant, right? Both of these look like completely reasonable normal sequences, double-stranded DNA with base pairing all the way through. So in fact, we don't really know which one is the mutant, so maybe it makes sense when we have a difference to mark the two different alleles. So we'll mark one with the hash to the left and one with the hash to the right. Okay, so then if we include that, that would mean that once we have this mutation here, that we can indicate the two alleles, this one versus these 3. So I just used this word alleles. Hopefully you're getting accustomed to it. That's a difference, right? That these are basically the same except for mutations. And so now we have these differences that we're tracking. And so this cell is giving rise to 2 cells which will inherit that variant. And these other cells will give rise to these other cells which will have the other variant. And in the next generation, the same will be the case. The 4 cells that are descendant from this mutant cell will have the left hash. And then the 12 cells that are descendant from other cells will have the right hash. Right? So we have this difference here that's been propagated through time. So we could also have a mutation at another site. So this would be another site in a different gene on the same chromosome. And that will be inherited. And so now we have two different variants at this site, this chromosomal location. And then we can do one on the blue chromosome as well, where we have a mutation arising, and now we have two variants, again left and right, with the blue. Okay, so this means that within the population, within the modern population, we have these different kinds of chromosomes, right? We have different kinds of black ones, and we have different kinds of blue ones. So among these, we have some pairs, like these two chromosomes, are the same. You can see they're identical. There are no mutational differences between them. And then we have other chromosomes that, just like our starting chromosomes, are different, right? This blue chromosome still has NOVA and RPG, and this black chromosome still has lactase and the pigmentation gene, right? They're different chromosomes. So we've got some that are the same, we've got some that are different, and then we've got some that are kind of the same but different, right? They have this difference, and that difference is due to mutations that have occurred in the history of these chromosomes. But we also have blue pairs that are actually the same, right? So we have blue pairs that are either slightly different because of mutation or blue pairs that are the same. And collectively, altogether, every pair of blue chromosomes here we can call homologous, right? So this pair that are identical, or this pair that are the same except for these mutations, this is what we mean by homologous. And so let's follow this through. If we then have these two haploid cells, which fuse to give rise to a diploid cell, so now we see that they've got two blue homologous chromosomes and two black homologous chromosomes. And now when this diploid undergoes meiosis, what we see is that the black chromosomes are lined up with each other, and the blue ones are lined up with each other. That is, we see pairing of homologous chromosomes that are the same except by mutation. So hopefully this clears up the concept of homology. Thanks a lot.